Hi guys, we're back with Concept 16.2, the second concept out of Chapter 16. So now we know, or the scientists at the time know, again this would have been about the 1950s, mid-50s through the early 60s. We now know that DNA is the molecule that is the carrier of genetic information, that all our genes are located on this DNA molecule. And this DNA molecule, apparently somehow the information is passed from parent to offspring. So DNA is a type of a long chain macromolecule made up of nucleic acids, made up of 4 possible different nitrogenous bases, and we talked about those previously, the cytosine, guanine, adenine, and thymine. And then we know that that's attached to a 5-carbon sugar and then to a phosphate group. We know that those chains are arranged with a certain directionality, what we call from 5' to 3' position, and that's based on the carbon in the sugar ring that we have numbered and what we're talking about. So we also now know that the DNA is in the shape of a three-dimensional shape of a double helix, and that was based on Watson and Crick and their information along with the X-ray crystallography of other people, other work, Rosalind Franklin and other scientists. So concept 16.2 is now going to take what we know about DNA and its structure and get down to how does it replicate, how does it divide or copy itself and prepare itself to go into multiple copies of the cells. So you could be talking about something like mitosis, where your skin cells are dividing, and every one of those skin cells need a copy of that DNA so they have the instruction code to know what to do. Or you could be talking about the formation of gametes. We could be talking about meiosis. How does that process occur, where DNA from the parents gets into the gametes and then combines to make a new offspring? So 16.2 is all about that replication process, and it says many proteins work together in DNA replication and repair. I'd love to tell you that DNA replication is a very simple process so we can finish it in 5 minutes, but as usual in biology, it's not so simple. It is fairly complicated, but I wouldn't say it's so complex as to be confusing. There are just a number of steps that are kind of going on at the same time that we need to be aware of. So we're going to talk about both DNA replication, which is the copying process, and then we're also going to talk about repair and upkeep of DNA, because that is a very important process as well. As we've talked about in this biology class over and over and over again, structure and function kind of go together. So if you know the structure of an organ or the structure of a molecule, you can ascertain to some degree a bit about its function and its purpose and how it works. And the same thing applies to DNA. The relationship between structure and function is explained or manifest, made viewable in the double helix shape. Watson and Crick noted that specific bases pair up together. And this is important because it suggests a way that DNA can copy itself. If you know that A always binds to T and G always binds to C, then you can create a copy of either strand by using the opposite strand. So at a portion in the strand where you have an A, you know the opposite strand will have a T at that same portion. At a portion in the strand where you have a G, you know the opposite strand will have a C at that same position. So because certain bases, nitrogenous bases, pair up together and only with that other base, this creates basically a copy mechanism. And you can have a single side of the strand and you could make a copy of the other side because you know what's going to pair together. The basics of DNA replication are this: because we have a 2-strand molecule and because the 2 strands are complementary, each strand can serve as a template for the other. And in DNA replication, that's exactly what happens. The parent molecule, the parent DNA molecule, basically unwinds and comes apart in the middle, and then the 2 new daughter strands are built based on the base pairing rules that we've already discussed. Here's a good animation that you can hit right click and play, and it shows you a very simplified version of the replication process. But I'll also kind of show it to you in some slides coming up in just a moment. So let's start with a parent DNA. Here's the parent molecule in DNA. It is a long chain molecule. Of course, this is a very simplified version of it. So here we have the sugar phosphate backbone running one direction. Here's the sugar phosphate backbone running the opposite direction. They're anti-parallel. In the middle, we have these nitrogenous bases. A always forms a pair with T. C always couples with G. And so we know wherever we see an A, the opposite side will be a T. Wherever we see a T, the opposite side will be A. Same with C and G. So here's our parent molecule. Basics of replication are that this molecule will unzip or separate down the center, and then these two strands can each form a new strand of DNA using this as a template. So each daughter DNA molecule consists of one of the parental strands plus one brand new strand that was created to match those bases. So in the two new cells, you have kind of a mix of the old DNA from the parent and the new strand of DNA on the other side combined with that. Recall that we call this a semi-conservative model because a portion of the DNA is conserved and a portion is brand new. So this would be called the semi-conservative model of DNA replication. Watson and Crick's semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule will have one old strand, that's the part that we say is conserved from the parent molecule, and one newly made strand. Early on, there were some other possibilities that scientists considered. There were some other competing models. Either there was one called the conservative model, and we'll take a look at a picture which helps. And a conservative model is where the two parent strands rejoin together. So you would have two strands of DNA. One is the old parent strands, and one is made up of two new strands. And then you also have something called the dispersive model, where each strand of DNA is a combination, a mix of both old and new. And in the beginning, when we weren't sure, all three of these models were possible. So scientists considered a semi-conservative model, which that's the one that it has turned out to be, and they also considered what they call a conservative and a dispersive model. And so until we could rule out two of the models, you kind of have to consider that all three are possibilities. Here's a picture or an illustration that kind of tells you a little bit more about them. The conservative model, and again, they didn't know exactly what the process might be. This was just an early model. The conservative model would be one in which your two strands of DNA end up with the parent strand back together and the other two new strands combined. And then a second replication would be, again, parent strand back together, and then this would make two new strands again. So this would be what we call a conservative model, where the parent strands of DNA always come back together and the other strands are brand new. The semi-conservative model, which is the one that it has turned out to be, ends up with a combination. Say if you had two replications, first replication would be 50-50, old parental DNA and the new strand, and then if that replicated again, one of those would be still 50-50, and then one of them would be completely new. So 50% of the material in this case is parental and 50% is new. And then in the dispersive model, which turned out not to be the case, they weren't sure how this would work, but they thought perhaps every strand of DNA was a combination of both old and new, and that would continue. So in a second replication or third or fourth, that every strand would be a combination of both old and new DNA. So this is the model, semi-conservative model, the one that won out, and the reason it won out is because it goes best with the facts of experimentation that they did later on. So we'll talk about how they came to understand that the semi-conservative model was the correct one. In the late 1950s, California Institute of Technology scientists Matthew Michelson and Franklin Stahl, they developed a very elegant experiment for determining which one of these three possibilities was correct. So remember, at the time, we still weren't sure what was the correct model of DNA replication. So they developed an experiment by using the E. coli bacteria. The E. coli bacteria is one that's often used in these cases because it has a fairly short or small genome. The E. coli bacteria only have one single chromosome. Compare that to the 46 that humans have. So for a lot of different reasons, E. coli is very good in genetic studies because it's quite more simple to investigate and look at with its single chromosome and things like that. So here's what they did. They took E. coli bacteria and they grew it in a medium, a growing material of different nitrogen isotopes. And they had two different types of nitrogen that they were using in this particular case. First, they grew the E. coli in a medium that contained a heavier isotope of nitrogen called nitrogen 15. So a slightly heavier version, a slightly higher atomic mass called nitrogen-15. And after they let the E. coli grow in that material for a while, then they let it replicate for a few times, and then they transferred it to a normal nitrogen-14, which would be a lighter isotope, a more normal one that you would expect. And so basically the E. coli incorporated that nitrogen into their DNA, because DNA does contain nitrogen as one of its components. And so that differentiated the DNA into a lighter and a heavier form using the two nitrogens that came from the medium that they grew in. So basically the older nucleotide strands were made with the heavier isotope, and the new nucleotides were kind of created or labeled with the lighter isotope of nitrogen. After the first replication of DNA, they took a sample and they put it in a centrifuge to make the material separate out into the lighter and the heavier constituents. And that first replication, we'll see in just a moment how, but it eliminated the conservative model that we talked about. So in their first replication experiment, the conservative model got knocked out as a possibility. And then they allowed the bacteria to go through a second replication of DNA. And the second replication, again, they took that material and they spun it in a centrifuge. That produced, again, this will be clear in just a moment, both light and hybrid DNA, and that eliminated the dispersive model. And so what was left was the semi-conservative model, which turned out to be the correct one. Now this is made much clearer if we look at some pictures and illustrations. So let's take a look at some illustrations. And you could also look on page 312 of your textbook where the same illustration is, and it'll be a lot more clear than just saying it in words. So here they have the two vials, and in this vial was a culture medium that was made up with the heavy isotope of nitrogen. And they allowed the bacteria to grow in this isotope for quite some time, and then they transferred it over to the medium with the lighter nitrogen isotope. Then, after the first DNA sample went through the centrifuge, after the first replication, this is what they got. They got different bands of material with kind of a mixed band of the nitrogen DNA material in the middle. So this mixed band tells you that it's a combination of both the lighter and the heavier. Then, after the second replication, again, they took a sample and they spun that in the centrifuge, and they got two different bands. They got a light band, a less dense band with the lighter nitrogen, and a more dense band with the heavier nitrogen. So these two these two pieces of information, these two experimental samples, gave us the information we needed to pinpoint the particular model. So let's take a look. So in the first replication, here are our three models, and here are the predictions. If we have a conservative model, you would have, remember, one strand of the DNA that's made up of all the parental DNA and one brand new strand. So this should have, the older DNA should have been labeled with the heavier nitrogen, and the brand new strand of DNA would have come from the lighter nitrogen. So if this is the case, if the conservative model was correct, you would have ended up with basically two bands. And that's not what we got. We got just a single mixed band, notice right there in the middle. We didn't get a light, less dense band on top and a more dense band on the bottom. We got just a single mixed band. So that means the conservative model is not the way that it worked. Down here, the semi-conservative model, remember, would have half parental and half new. Well, that would give you one band in the middle. So this one still could be the case. The dispersive model would be made up of a mixture of old and new, and that would give you the same results. So after the first replication, we can knock out the conservative model, but we still don't know. It could either be the semi-conservative or the dispersive model. So now we're going to go for the second replication, which was done after they moved the bacteria into the lighter isotope. So again, conservative model, it would come out with basically the bands that you see here, with a band light toward the top, heavy toward the bottom. Well, that's not what they got. They got a band light toward the top, and they got a band in the middle. So again, that knocks out the conservative model. If you needed to knock it out twice, this knocks it out twice. The dispersive model. The dispersive model, again, would be a combination of both light and heavy. So you would expect one single band, maybe toward the middle or the middle top. That's not what they got. They got two separate bands, so that knocks out the dispersive model. Let's take a look at the semi-conservative. again, based on our previous model, we would have now two replications. One would be a hybrid mix of old and new, and the second replication would be all new. So you would expect one line of material toward the middle and one line less dense toward the top. And that's exactly what they got. So these two different steps, two different replications, helped knock out the different models until only one was left, and it was the semi-conservative model. So what that tells us is when DNA replicates, it uses this semi-conservative model to where in every new strand of DNA in the first replication, it's a mix of one strand of parental and one strand of new. And that is going to be shown in more detail in just a moment. In the second replication, you would have one portion be still a mix, and then one portion would be brand new. And that's what both phases of their experiment showed. So this was a very well-known experiment done by the California Institute of Technology, by Michelson and Stahl, and it's oftentimes referred to in genetics. So now we know the basics. We know that the DNA strand basically unwinds, unzips, and both parental strands are used as a template for the other half of a brand new strand to be created. So let's take a closer look at this, because the copying of the DNA is pretty amazing in both its speed and its accuracy. And that's, if you think about it, each one of your cells has 46 DNA molecules in the nucleus, and each one of these molecules is a long double helical shaped molecule, each one per chromosome, made up of about 6 billion nucleotide pairs. If we were to print out just the one letter symbol for these bases, so not write out adenine, cytosine, guanine, but just print A, G, C, T. If we were just to print out the one letter symbols for these bases in the same font that you find in your textbook, that size, it would fill 1,200 books as thick as your biology textbook. That would be just the letters that represent the DNA nucleotides. The nucleotides that make up the DNA in a single cell inside your body. And it's a fairly complex process. Like I said, there are more than, we're going to find out, a dozen different protein enzymes and other proteins that participate in DNA replication. And the amazing thing is that this is a fairly fast process. For all that information to be copied, it takes your cells only a few hours to replicate this DNA. And they also do so with a very, very low error rate. There's only about one error in the DNA sequence per 10 billion nucleotides. That's pretty good. So we're going to look at how this replication process takes place, and we're also going to look at how can it be so error-free. It's important that it is error-free, but we're going to talk about what the body does to try to maintain this error-free replication process. I've already mentioned the E. coli bacteria, and much of what we know about DNA replication we have learned by studying the process in the E. coli bacteria. Remember, I say the E. coli has a very simple genome. It only has one chromosome. Its DNA is different from ours. It has one strand of DNA that's in a circular shape. So there are some things that are quite a bit different between a bacteria, of course, and a eukaryote like us. But a lot of what we've learned in the E. coli applies to our cells as well. So while we're going to be talking about the E. coli, we're also going to talk about how that information applies to eukaryotes like plants and mammals and birds and fish as well. The whole copying or replication process begins at particular areas along the DNA molecule called an origin of replication. And this is where the two DNA strands begin to unwind from each other and separate, and they open up what's called a replication bubble. If we're talking about a simple bacteria like E. coli, they have a single site or origin of replication, and then that goes along in both directions along the whole DNA circular molecule of theirs. In eukaryotes like us and other mammals, there's going to be many hundreds or sometimes even thousands of these little origins of replication that pop up. So all along our DNA molecule, there will be various spots where the molecule unwinds from itself and begins to separate and form these bubbles of replication. And then the replication process actually begins, and it proceeds in both directions. So it will go both left and right all the way through until the entire replication process is complete. Here's a great little animation that kind of talks about the origin of replication and how the replication bubble begins to form. So you can hit right click and play and see a quick little video. Here we have a great illustration and some pictures kind of showing how the E. coli cell is like the eukaryotic cell of a human or a mammal and how they're different. So in an E. coli cell, it's very simple. They have a single chromosome, a single DNA molecule, and it's in a circular shape. They're going to have a single point of origination, a point of replication that's going to open up a replication bubble, and then you're going to have what's called a replication fork. That's the Y shape where the bubble meets the still unwinding molecule, and that's going to continue in both directions. So both around toward the left and around toward the right, that replication bubble widens and opens, and they are making the new section of the DNA inside this replication bubble until that goes all the way completely around. You end up with basically a complete copy, and then that gets subdivided into the two daughter E. coli bacteria cells, into the two new daughter cells. So then you have two exact copies of the DNA. In a eukaryotic cell, it's a little bit more complex than that. We have multiple origins of replication, and so here we have the double-strand DNA molecule. They begin to separate in various different spots, and that forms what we call a replication bubble, and that's going to proceed both directions, left and right, making the light blue that you see inside here is the new strand of DNA. When we're done with that, we're going to have two strands of DNA. We're going to have one, the dark blue is still the parental original strand, and the light blue is the newly created daughter side of that double strand. And here you have a picture showing some areas of replication, some bubbles that are opening up in that DNA molecule. Here's some basic vocabulary that we need to add to, and of course, anytime we're talking about vocabulary, if you're like me, flashcards like Quizlet and that type of thing are very helpful, but there's some terms that we need to know because when we show pictures and illustrations or we watch the videos, they're going to use this terminology. At the end of the replication bubble is something called the replication fork. That's the Y-shaped area where the new DNA strands are being created and elongating, and I'll point this out in just a moment on a picture. Then we have some enzymes that we're going to need to know. One of the enzymes is called helicase. If you recall, the DNA molecule is a twisted ladder shape. The DNA has to be unwound or untwisted before it can be replicated. So the enzyme helicase does the untwisting of that double helix right there at the replication fork. Then we have some other proteins and enzymes that are hard at work. One of them is called single-strand binding protein. The single-strand binding protein is basically what keeps the two strands of DNA separate from each other because they have a tendency to want to go back to where they were. So the single-strand binding protein helps keep the DNA strands separated from each other and keeps them from rebonding. And then we have another enzyme or another protein molecule called topoisomerases. And that's a tongue twister. Topoisomerase. Topoisomerase corrects what we might consider the overwinding ahead of the replication fork. So imagine you have, say, two strands of yarn that are wound together in a helical shape, and now you are pulling those two strands apart. Well, right where they meet together and are twisted, they become tighter and tighter, more twisted together, and we would call that overwinding. So this topoisomerase helps fix that by breaking, swiveling, and rejoining the DNA to kind of relieve the strain there. So let's take a look at these vocabulary words along with a picture, and that'll help us kind of put them in our memory a little bit better. So here we have the DNA molecule, and in this case it's already been unwound, so they're not showing it wound, in other words. Here we have the one enzyme that we call helicase. Helicase is the one that's doing the unwinding or the untwisting of the double helix, and it does that right there at the replication fork. Here we have the single-strand binding proteins. They are the ones that help keep the two strands separate and apart from each other so they don't kind of go back together. Here we have the topoisomerase. The topoisomerase is the one that helps disconnect and reconnect the DNA here, because otherwise this would be all twisted up and it would become more tighter and tighter twisted as this portion was unzipped. And then we're going to talk about the primase coming up. We didn't discuss that one yet. But next on our list is the primase and some of the other enzymes that start the whole replication copying process. Oh, and one more. I guess we could also talk about the replication fork. So when I say replication fork, see how it kind of looks like the shape of a Y if you look at the DNA here? Right here, this is the replication fork. So this area where the DNA has been pulled apart and the whole copying process is about to begin right in here, this is the replication fork that we're talking about right here. We already mentioned that there's a number of enzymes that are kind of involved in this process, and the main one is called DNA polymerase. DNA polymerase is the enzyme that creates the new strand of DNA, but it's fairly complex because it has some limitations. DNA polymerase cannot start or initiate the synthesis of a polynucleotide, a new chain of nucleic acids, which that's what DNA is. It can only add nucleotides to the 3' end of an existing chain. So it kind of has a limitation here. It can't start a new chain. It can only add to an existing chain. So what has to happen is you have to create what's called a primer. And a primer is a short initial nucleotide strand that's made out of RNA. Remember, RNA is a different type of nucleic acid chain, ribonucleic acid. So you have to have what's called a primer made out of a little RNA chain. And the enzyme that makes this little primer is called primase. So let's go back and look at that picture. So here we have an enzyme called primase. And it's going to create a little short section of RNA called an RNA primer. And then from off of that primer, we can start to make the actual new strand of DNA. So you want to consider this short little strand of RNA, this primer, sort of as a signal or a starting line to show the DNA polymerase where to begin. Because the DNA polymerase can't start the chain from scratch. It has to add to something that already exists. So the RNA primer is created by primase, and primase can create its own little new RNA chain from scratch, and it adds RNA nucleotides one at a time using the DNA as the template. So if we come back here, the RNA primer, it knows what to put there because it's going to use this parental chain of DNA as its template. And it says, okay, well, here's an A, so we need this, and here's a G, so we need this. And it adds the correct RNA sequence based on this DNA chain, using it as a template. The RNA primer that it makes is fairly short. It's about 5 to 10 nucleotides long, and the 3' end serves kind of as the starting point for the chain for the new DNA strand. So then the DNA polymerase can say, oh, okay, well, here's a beginning chain, and I can add on to it. So as we mentioned, the enzyme that actually creates a new strand of DNA, it's called DNA polymerase. It catalyzes the elongation of the new DNA right there at that replication fork. Most DNA polymerases require some sort of primer or starting point, and in this case, that's a little short section of RNA made by primase. And the rate of elongation is about 500 nucleotides per second in bacteria and about 50 per second in human cells. So the bacteria can actually replicate its DNA faster than the human cell can, but about 50 nucleotides per second, which is actually a pretty quick process. Like we said, the entire DNA of a cell, 46 chromosomes, can be replicated in just a matter of hours in a human cell. If we discuss a little bit about the chemistry of the DNA molecule, each nucleotide, remember this is a chain of nucleotides that we're creating. Each nucleotide that is added to a growing DNA strand is a nucleoside triphosphate. And we'll take a look at a picture here in just a moment. The DATP is what supplies the adenine to DNA, and it's very similar to ATP. So there's a molecule called DATP that, if you'll remember, ATP, we've already discussed that, that's adenine triphosphate. Well, this molecule is called DATP, and that's the nucleotide that supplies the adenine to the DNA, and it's very similar to ATP. The only difference is the type of sugar. So just like there's RNA and DNA, and the only difference is a portion of the sugar, Well, here's a molecule called DATP, which is very similar to ATP. The only difference is the DATP has a deoxyribose sugar, while the ATP has a ribose sugar. As each monomer or chain, link of chain of DATP joins the DNA strand, it loses two phosphate groups as a molecule, what we call pyrophosphate. So let's take a look at kind of the chemical reaction that's going on here. So here we are building the chain. This was the original template from the parent DNA strand, and here we're building the new side of the DNA strand. Here's our sugar phosphate backbone, and here is one of the molecules we're going to add. Together, this is called a nucleoside triphosphate. It's made up of a nitrogenous base, thymine in this case, and then the ribose sugar, and then it's got 3 phosphate molecules stuck to the end here. And then when it gets added, this is basically a hydrolysis reaction. It's an exergonic reaction. The DNA polymerase is going to cause this hydrolysis reaction to occur. That's going to change this molecule slightly, and it's going to release 2 of the phosphates that were on the end as a what we call a pyrophosphate. So then when the molecule is attached, it's going to be the same, except now it only has the 1 phosphate instead of the 3 that it began with. So now we have the nitrogenous base, thymine, and this is the, I think I said ribose sugar a minute ago. It's deoxyribose sugar, and then the single phosphate, and that's going to then be connected to the next one, connected to the next one, connected to the next one. And again, if we look at it sort of with directionality, this is the 5' to 3', and you'll notice the other strand runs 5' to 3' in the reverse direction. So they're anti-parallel. So we could have like an arrow going this way, and this would then represent an arrow going this way. And again, you'll notice that only certain bases match up together. A is always with T, C is always with G. So if you know one, you know what the opposite side is going to be. So we know that this is what we call anti-parallel elongation. The 2 strands are parallel to each other, but they run in the opposite directions, and that is because of the structure of this double helix. But that also affects how we're going to copy these strands or replicate these strands, because remember I said DNA polymerases, they are obviously pretty cool. I mean, they are the enzyme that creates a new strand of DNA, but they have a couple of limitations. One of the limitations we already mentioned was they can't start a brand new chain all on their own. They have to have this RNA primer created by primase to get them started. Well, here's another limitation. The next limitation is that they can only add links to the chain in one direction. So DNA polymerases can only add nucleotides to the free 3' end of a growing strand. What that means is they only work really in one direction. So a new DNA strand can only get longer in the 5' to 3' direction. And that's going to be a bit of a problem. For one of the strands, it's what I would kind of call the easy side of replication. Along one of the template strands, the parental strands of DNA, the polymerase synthesizes what we call a leading strand. And so when you hear the phrase leading strand, I want you to think of kind of like the easy side here, because this strand is going in the proper direction. So it can start the replication process at the replication fork and just continue going down the line, no problem. So it's moving from the 5' to the 3' direction, and that is no problem whatsoever. As the fork widens, the replication just continues on down the line. So recall that the replication fork is that Y-shaped area where the DNA is unzipping. So I usually call the leading strand the easy side of replication. Now, if you want some more information about this, I highly suggest clicking on the YouTube link here. I believe, I can't remember, but I think this one is the Bozeman Science link. And it talks about DNA replication, the leading strand and the lagging strand. And as always, I suggest you go at this from a wide variety of angles. So read the textbook, listen to the vodcast, go watch the crash course videos, the Bozeman Science videos. If you look at it from multiple angles, it'll make a lot more sense. So we have 2 different strands. The one we're looking at first is called the leading strand. That's going to be the easy side, and you'll see why I say that in just a moment. Here's a great little video. Click on the animation, right click, hit play, and it talks about the leading strand, again, which is the one side of the strand that's kind of easier or less complicated to copy. Take a look in your textbook on page 315 for this illustration, which I think kind of clarifies a little bit. So here we have the DNA that is unzipping, or basically coming apart, and then we're going to create two new DNA strands along the way. So here we're moving from the 5' toward the 3', and that is what I would call like the forward direction or the easy direction. So this one up here is going to be called the leading strand. This is our leading strand that we're looking at. And we have the RNA primer that was created by the primase. And then we have one of the DNA polymerases that is creating the new section of DNA, the light blue that you see here. Now, as this DNA unzips, this DNA polymerase is just going to continue on down the chain, and it'll never have to stop. So here you see it's continuing on down. As this unzips further, we're making that new chain basically without stopping, no problem. So this is called the leading strand, and this is moving from the 5' toward the 3' direction, and the DNA polymerase works in that direction. So this is the easy part right here. Now let's take a look at the problem child. We're going to call the other strand the lagging strand. To elongate the other strand, it's going to be a bit more work because the DNA polymerase can't work in that direction away from the replication fork. So this is going to be problematic. So the lagging strand is basically created in small little chunks. It's synthesized as a series of little segments, and they're called Okazaki fragments. And that's named after the scientists that actually discovered them and named them. Happened to be a husband and wife team, last name Okazaki. So these little sections of DNA that have to be made for the lagging strand are called Okazaki fragments. And then those little sections have to be tied together, and that's done by a different enzyme called DNA ligase. Without looking at a video or a picture, it's kind of hard to understand how this works. So I was trying to come up with a model for how this might work. So let's imagine you have adding machine tape, you know, the long, thin rolls of tape that you get from an old adding machine. And imagine you have this long roll of paper, and you have a pen in your hand, but your pen can only mark from right to left. So your pen can only make a mark when it's moving from right to left. But your roll of adding tape is unspooling to the right. So you have a roll of adding tape, and it is unspooling or unfolding. It is rolling to the right. And you're supposed to make one long continuous line on this adding tape as it unspools toward the right. But your pen can only mark from right to left. Well, how do you do that? Well, you're going to have to let the spool unwind toward the right a little bit, and then you can mark that section from right to left. And then you're going to have to wait for the spool to unwind a little bit more to the right, and then you're going to have to jump over and draw another line from the right to the left. You're going to have to continue to do this, and then you're going to have to make sure your line segments that you've drawn, if they don't meet up exactly together, you're going to have to draw in a little joiner line to make sure they meet up together. So you can eventually get one single solid line down this whole unrolling tape of paper, but you're going to have to do it in short little segments because the paper is unwinding to the right, but your pen only works from right to left. That's very similar to how the lagging strand of DNA replication works. So your little segments that you have to draw are the Okazaki fragments, and the parts where you have to make them meet up to join to be a single continuous line, that's done in DNA replication by this enzyme called DNA ligase. Again, another helpful animation. I highly suggest you right-click and hit play so you can kind of understand what we're talking about, how the leading strand can work in one continuous long motion, and how the lagging strand has to be created in small little increments and then put back together by the DNA ligase. In this illustration, we have the DNA is basically unzipping or unwinding off to the left. But remember, our enzyme can only work from the 5' to the 3' direction. So in this case, our DNA is unzipping off to the left, but we can only work from left to right. So we have a little RNA primer that gets created, and then we can create a little section of DNA. And that's our Okazaki fragment. And then we have another little primer that can be created, And then we have some more DNA that gets created by the DNA ligase. And then we have to correct that. We have to connect those two together. So basically, we can eventually move, or overall we're moving toward the left, but we're having to do it in short increments to the right. And this is called the lagging strand. So instead of one smooth, continuous new daughter strand, we're having to make that new daughter strand in multiple small segments called Okazaki fragments. Here's another set of illustrations to kind of prove the point. So our DNA polymerase can only move in this direction in this illustration from left to right, from 5' to 3'. So we're going to create a little RNA primer. Then we're going to create a little strand of DNA. This is an Okazaki fragment. Then, as that unravels more, we have more space. We can create another little primer, and then we can create the second section of DNA moving from left to right here. Then we have to come along and we have to use DNA ligase to connect these two sections. So they are DNA, RNA primer, DNA, RNA primer. We have to fix that to make it one long, smooth, new daughter strand of DNA. So overall, we're moving to the left, but again, we're doing it in small increments of left to right segments of DNA. So here we have both strands in the same illustration. The top strand is the leading strand, and it is moving in the proper direction for the DNA polymerase. So it creates one new long strand of daughter-created DNA. The lagging strand, that's where we have the difficult work. We have to go this direction, create small pieces in this direction, and then add those together or bring those together. So the DNA is unzipping right here. The leading strand can move simply in that direction, but the lagging strand has to move the opposite direction in small fragments called Okazaki fragments that then have to be bound together by the DNA ligase. Again, another good animation showing kind of an overview of the entire DNA replication process. So right-click and hit play, and you can see kind of an overview of the whole process, including both the lagging strand and the leading strand. And at the end of this, we'll have two replications, two copies of the DNA, and they should be without error. If they're without error, two exact copies, one of the other. I do want to point out that in all the models we've been looking at, it kind of shows all these various enzymes kind of working separately and not really connected together. But in reality, what we usually have inside the cell is something called the DNA replication complex. And the complex is a big amalgam of all these proteins and enzymes that are kind of all stuck together and that work together. So they don't really act individually and separately from each other. They more tend to act in a big clump where a whole bunch of the processes are going on all at one time. And we were showing kind of the DNA staying still and the DNA replication machinery kind of moving along like the DNA was a track and the enzymes were a little train moving along. Well, in actuality, a lot of experiments show that it doesn't really work that way, that the DNA actually is the one that is doing the moving, that the replication complex, these proteins and enzymes, kind of tend to stay perhaps attached to certain parts within the cell, within the nucleus, and then the DNA kind of threads through them. So recent studies show or support a model with the DNA polymerase molecules kind of reeling the DNA in and pulling it through and kind of spitting out and extruding the newly made daughter DNA out on the other side. And they even show the lagging strand kind of making a double loop back through the complex to kind of make these short Okazaki fragments and then connect them together. So some of our more modern experiments kind of show a DNA complex that looks more like this, where all these proteins and enzymes tend to be stuck together in one large machine. You could kind of think of it as a big, large machine. And here we're unwinding the parental DNA. This would be our leading strand, so it's going from the 5' to 3' direction, and you've got just a continuous new chain of daughter DNA being made on this side, and spitting out of this end is the combination of one parental and one daughter strand. And then on this side, we have the lagging strand, which, remember, can't work in that direction. It has to work kind of backwards And so you kind of have it working backwards in small sections, and it might make this double loop back through the DNA replication complex, and then it's being tied together. These little segments, Okazaki fragments, are being tied together down here at the end by the DNA ligase. So coming out, we end up with two strands of DNA. Both of them have one parent strand and one daughter strand, but it's kind of a bit more complex process for the lagging strand.